Welcome back to the A-Level Cookbook. Today we're going to be going through the third paper of the June 2020 series of AQA A-Level Biology. Guppies are small fish. Female guppies are dull in color. Male guppies can be bright or dull in color. Scientists investigated the effect of the female brain size and on choosing a mate. They used laboratory-bred female guppies with large brains and with small brains, and they set up a fish tank here. So we've got a bright male, a dull male, a female, and we've got a transparent barrier between the two. They observed each female for 10 minutes and recorded which male they were attracted towards. They repeated this with 45 large brain females and 45 small brain females. Suggest three possible limitations of this investigation. They're using fish that were bred in a laboratory to test this hypothesis. So there might be a behavioral element involved. Maybe in the natural environment in the wild, that might affect their behaviors. So lab raised guppies may not behave the same as in the wild. That's one limitation we can spot already. The next thing is, well, we've got these transparent barriers in between. Some courtship behaviors might require interactions between the two organisms. So they might actually have to physically touch or do something like that. And these guppies might also be involved as well. So that might be another thing as well. So the transparent barrier may not allow normal interaction. So you can see that they've done this for 10 minutes, and 10 minutes might not be enough time to actually make a decision. You know, if somebody's going on a date, maybe 10 minutes isn't enough time to decide, yeah, this is the love of my life. You know, maybe they might take more time. Or some might be content within 10 minutes. I don't judge. So that's another thing as well, is 10 minutes be enough time for the female to make a choice. Guppies with large brains are better identifying predators. The scientists found that only female guppies with large brains were attracted to male guppies that are bright in color. Suggest and explain the advantage of this behavior to the population of guppies. So what you've got is you've got a big brain guppy liking a colorful male. They're going to have kids and they're going to have a different proportion of big brained and colored and dulls. But then more big brains are going to like the colored ones. So they're going to have more and more children and fewer dulls because the dulls aren't getting any action. Eventually, what happens is you're left with a population full of loads and loads of big brains and loads of, colorful fish, but very little dulls. And why is that good? Well, so then based on this, we can say that we've got lots of big brains and lots of colors. So a lot more colors means more big brains, which means they're better at identifying predators. So now we need to translate this into, the actual mark scheme words. I would start by saying females with large brains would mate with males that are bright in color. So females with large brains mate more with bright males. They're going to produce offspring that are big brain, colorful, dark, etc. Blah blah blah. But more of them are going to be bright in color because there's more. They're going to be going for more of them that are in bright in color. So then the offspring are more likely to be bright. As a consequence, the big brain females are going to go for these increased numbers of colorful offspring. So the bright offspring attract big brained females, and then obviously eventually the population will have fewer and fewer dull and more big brain and colorful ones, and that is evolution. That's a change in allele frequency. So the population eventually has more. I'm gonna write BB for short. Big brain plus colorful. Obviously, in your exams, you write the full thing out. So they are better at identifying. Identifying predators and surviving. That's the advantage. Okay, so describe how the behavior of female guppies can lead to sympatric speciation. So allopatric speciation is where you have geographical isolation, stopping reproduction between the two populations. So there's a big river or mountains or something in between them, so they can't actually mate. Sympatric speciation is where there is no geographical isolation and something else is stopping the two from mixing. So the female guppies with big brains only go for large brain guppies, meaning that the duller, darker colored guppies don't get any of the action. So they are reproductively isolated, but there's no barrier in between. So that makes it sympatric speciation. So I'd start by clarifying your understanding of what sympatric speciation is. So there is no geographical barrier. And what actually happens is, is that the duller, guppies are reproductively isolated, as big brain females only mate with bright guppies. So this is speciation they're talking about. So for speciation to occur, you need to have so much difference in the two populations that they become different species. So what changes? The allele frequencies has to change. So you've got changes in allele frequencies, but you have so many that they can't actually breed to produce fertile offspring, and that makes them separate species. So many changes in allele frequencies, they cannot breed to produce fertile. Another example of sympatric speciation may be something like an organism behaves a bit differently, still able to reproduce and produce fertile offspring to start with, but because it behaves differently, maybe the courtship behavior is different, it gets reproductively isolated, no one wants to mate with it. Eventually, what happens is that gets separated and separated and separated off down the generations. If it's able to mate with other individuals with that different behavior, and eventually the two diverge and become different offspring. So, in northern India, there's a conflict of interests between farmers of livestock, such as cows, and people trying to conserve ibex, a wild goat. When livestock are given extra food, their populations can grow too large and compete with the ibex. What is the type of competition between the two is inter-specific, between species competition. Intra means within the same. Okay, so it says here, livestock will outcompete ibex if they're in the same habitat or eat a similar diet. Scientists investigated this conflict of interest. Table 1 shows some of these findings. So we've got the type of livestock here, cow, horse, yak. Difference between livestock food and ibex food, 1, 0.5, and 0. And a score of 0 indicates that the food or habitat is the same. So cows have a massively different type of food compared to the yak, who eats nearly the same thing. The difference between livestock and ibex habitat uses the same principle. So the horse has like nearly the exact same habitat. Cow has a relatively different habitat, and yak has like a pretty damn different thing. So the higher the number, the more different they are. So it says here there must be a balance between the need for conservation of the ibex and the need for the farmer to keep livestock. This makes sense because these farmers make their living out of these livestock, such as cow, but these ibex are getting in the way of it. But they don't want to get rid of all the ibex because that wipes out an entire species. But they also don't want to stop selling their livestock. So using all the information, suggested explain 3 actions the farmers could take to achieve this balance. So one thing you could do is control the amount of cow, horse, and yak they have, and ibex. So if you have loads of cow, horses, and yaks, like so many, they're going to start competing with the ibex for food. If you have a relatively small number, you've got enough food to supply for them. So you could start off by saying something like that. So small numbers of livestock, which kind of summarizes all 3 of those. So no competition occurs as the population isn't large enough. I'm writing pop for sure. Make sure you write the full thing. If the population is relatively tiny, there's not really going to be much of a problem. Now, let's look at the actual numbers. So the horse has pretty much the exact same habitat as the ibex. So I would say, maybe not farm horses near ibexes because they have very similar habitats. But as well as that, 0.5, that's pretty similar food. So actually, the horse is probably one of the worst things you could have near these ibex because their difference is very small in both food and the habitat. So avoid farming horses near ibex because food and habitat, food sources I should say, are nearly the same. They're probably the worst 2 to keep next to each other. So as well as that, what could they do? Well, you could farm yaks. Although they have the exact same food supply according to this table, the difference in habitats is quite different. So you could put them in 2 very different locations. So you could farm yaks in different habitats, even though they eat the same food. You could also suggest things like farming cows, for example. Their food supply is relatively different, and also their habitat's different. Or you could also say that these are farmers, so they could try considering, growing crops instead of animals and livestock, so there's no competition. But I don't feel too good with that answer personally, because, this is their job. Telling somebody, hey, mate, can you grow crops instead? Probably not the wisest thing. So in this mark scheme, it allows it, but it's better to sort of use the data in the question in exam questions like these. In Europe, viruses have infected a large number of frogs of different species. The viruses are closely related and all belong to the ranavirus group. Previously, the viruses infected only one species of frog. Suggest and explain how the viruses are able to infect other species of frogs. So you guys need to have an appreciation of how viruses roughly work. You guys do HIV in A level. How viruses roughly work is that they have a sort of they have like an attachment protein that is complementary to a cell surface membrane receptor. So what it does is it binds to this complementary receptor, the capsid goes in, and it releases all of its gunk. So that's probably what's happened here, is that we've got different species of frog with all these different receptors, etc. And what's probably happened is that this virus has somehow managed to change its attachment proteins. Proteins are determined by their tertiary structure, which is determined by their secondary and primary structure. The primary structure is the amino acid sequence, which is determined by the genes you have. So you've got this question is testing you working backwards and appreciating that. If you have a change in the genetic sequence, that is a mutation. So what's happened is the virus has mutated, and the proteins that it makes, the attachment proteins, have changed. This means that it can bind to the receptors of other species. So I would say here mutations in the viral DNA, RNA, or you could say genome, that kind of encompasses all of them, leads to altered or different, I should have just, sorry, leads to different tertiary structures in their attachment proteins. So it allows it to be complementary, as in allows the proteins to be complementary to receptors of different species. As a general principle, if a pathogen's able to infect lots of different things, it's probably because the receptors have tweaked. And the receptors are proteins. Proteins are determined by the genome. So mutations change the protein structure because they change the genome and they change the protein as a result. Name two techniques scientists have used when analyzing viral DNA to determine that they were closely related. So there's lots of different things you can do. You can do genome sequences, so you can look at the entire sequence, ATCG. Name two techniques scientists have used when analyzing viral DNA to determine that they were closely related. So there's lots of different things you can do. You can do genome sequences, so you can look at the entire sequence, ATCG, blah blah blah, and compare the two. You could do gel electrophoresis, so you've got this ladder of the different things, and you've got your organisms, and you can see how many of them match. You could do DNA fingerprinting as well, and the polymerase chain reaction. So any of those answers, really. I'm just going to write two here just for the sake of completeness. So electrophoresis and genome sequencing. Determining the genome of the viruses could allow scientists to develop a vaccine. Explain how. So we've mentioned up here, and the questions hinted at it here as well, is that viruses work by using different attachment proteins. They attach to a protein on a cell surface membrane and it allows it to enter the cell and cause infection. And we said, and have established in the parts before, saying that the protein is determined by the genetic code present. So if we determine the genome, we can then work out the proteome and then we can work out what potential antigens or attachment proteins the virus has. And then we can make vaccines to target those. So what we would say is we can identify the proteome and we can identify potential antigens to use in a vaccine. Okay, so describe how the B lymphocytes of a frog would respond to vaccination against rhinovirus. You can assume that the B lymphocytes of a frog react in the same way as the B lymphocytes of a human. Do not include details of the cellular response into your answer. So, just as a quick summary, the cellular response is all to do with phagocytes interacting with T cells, so engulfing and digesting the pathogen, expressing the antigens on its cell surface membrane, and then saying to a T cell, "Oh, mate, something's going wrong in the body." Everything beyond that point is your B cell response. So, how they work is that B cells bind to complementary antigens and they divide by mitosis to form identical clones known as plasma cells. These plasma cells all make monoclonal antibodies. So, they make antibodies of the exact same type, and then they eventually go on to form memory cells as well. So, that's pretty much your answer. So B cell binds to complementary antigen. I'm going to write AG for short, but in your exam, you write the whole thing. The B cells divide by mitosis to make clones called plasma cells. Plasma cells produce monoclonal antibodies. So I'm going to write AB for short. You write the full thing. And then they also form memory cells as well. So the B cells form memory cells. Whenever they're asking about B cell responses in exams, always make sure to sort of use this formula when it comes to writing about it. So B cells always do this. They always bind to a complementary antigen or a signal from a T cell, a chemical signal from a T cell. And then what they do is they divide by mitosis to make clones called plasma cells. And then those plasma cells make monoclonal antibodies. A lot of people miss this part saying divide by mitosis and plasma cells. The reason for this is because we have a huge number of different B cells, all with different receptors and different antibodies that they make. So we want to make sure we get the right ones. So what happens is they bind to a complementary antigen and they undergo clonal selection and they make loads and loads and loads of the exact same one, all of which make the exact same antibody. People make the mistake of saying B cells make loads of monoclonal antibodies. That's kind of wrong because you're making the assumption that you're making monoclonal antibodies from all kinds of different B cells, which would be a bit pointless in an immune response because you want it to be specific to that one pathogen. Table 2 shows information about two types of medicine. Name of medicine, so we've got aspirin and paracetamol. We've got 300 milligrams of aspirin, 500 milligrams of paracetamol. The maximum dose allowed is 0.6 grams every 4 hours, 1 gram every 6 hours, and the mass of sodium in one tablet is 0.15 grams and 0.43. A journalist studied the table and data too and made the following suggestion. If an adult takes the maximum number of tablets allowed for either of the medicines, the person would have more than the daily allowance of sodium. The recommended daily allowance for sodium in an adult human is 2.4 grams per day. Is this statement true? And use suitable calculations. So for aspirin, one tablet is 300 milligrams, which is the same as 0.3 grams. You divide by 1,000. You're allowed 0.6 grams every 4 hours, and this is in a day. So the amount of times you can take this tablet would be 24 over 4, which is 6 times a day. So the max you're allowed would be 0.6 times 6, which gives you 3.6 grams. So to work out how many tablets of aspirin you're allowed to take, you would divide this by the amount of aspirin in one tab. So you would do 3.6 divided by 0.3, and that gives you 12. So you're allowed 12 tablets maximum in a day. The mass of sodium in one tablet is 0.15 grams. So in 12 of them, it would be 12 times 0.15, and that gives you 1.8 grams, which is less than the recommended daily allowance. So they're allowed to take the maximum for aspirin. So the statement is not true for aspirin. So for paracetamol, it's the same thing. Okay. So for paracetamol, one tab is 500 milligrams, which is 0.5 grams. The maximum they're allowed 1 gram every 6 hours. There's 24 hours in a day. 4 times 6 is 24. So it would be 1 gram times 4, which is 4 grams. So again, the number of tablets they can have is the maximum divided by the the amount in one tablet. So 4 grams divided by 0.5 is 8 tablets. The amount of sodium in one of these tablets is 0.43. So 8 times 0.43 equals 8 times 0.43 gives you 3.44, which is higher than that 2.4 that the journalist was stating. Therefore, the statement is true. Doctors investigated the link between high sodium concentration in medicines and hypertension. They analyzed medical records of patients, and 1,292,337 of these patients had taken medicines containing high sodium concentrations. Each of these patients were paired with a patient from a control group. Give two factors that would be the same for each pair of patients and one for that would have been different. So, generally, with these kinds of clinically trially questions, you want to sort of control different factors. You might know from the media and stuff that like things like diet, exercise, health, other medications, and ethnicity play a role in blood pressure as well. It's not just salt. So really, things you'd keep the same are things like, age, sex. You could also say, medications that they take, their ethnicity, diet, exercise, health, and stuff like that. The different factor though is that you are investigating sodium on hypertension. So if you're going to compare it with a control group, you need to take sodium out. You're testing one group with sodium, the other one without. So in this case, the different factor would be there would be no sodium in the other pill that this other group is taking. So in the same medicine. Because you want to keep absolutely everything the same, you just want to take the sodium out so you can see if sodium is causing a problem. Okay, so doctors found that 4.73% of the patients who'd taken medicines containing high sodium concentration had hypertension. There were 7.18 times fewer control patients that with hypertension. So calculate how many of them had hypertension. So this number of patients took medications with high amounts of sodium. So what we're going to do is we're going to work out that 4.73. So you do 4.73 divided by 100 to convert it back to a decimal times 1292337, which gives you 61127.54. And you can't have zero point 0.5 of a person, but I like to keep numbers in decimals as much as I can until the end. There were 7.18 times fewer control patients with hypertension. So of these people, there were 7.18 times fewer control patients with hypertension. So all you do is you take this number, 61127.54, and you divide it by 7.18, and that gives you 8513.5. Now, you obviously can't have half of a person, so now you've got to sort of round it up. So 8514, or 8513 is fine as well. Either's fine. A high concentration of sodium in the blood can affect water potential and cause hypertension. Use your knowledge of water potential to suggest how high sodium concentrations in the medicine can affect blood volume. In your blood, you've got water, you've got proteins, you've got blah, 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 right? If you have more sodium in the blood, the water potential of the blood is going to decrease. So water wants to move into the blood, into your blood vessels via osmosis. So you've got more water going into your blood vessels, increasing the blood volume, which in turn increases your blood pressure. So you've got this blood vessel chilling here, you've got hella sodium, and you've got like some amount of blood in there. If the water moves in, you're putting more and more water into this blood vessel and it increases the pressure. So sodium ions lower the water potential, which I'm going to write as WP. You write the whole thing. Water moves into blood vessels via osmosis from surrounding cells, and this increases the blood volume. If you increase the blood volume, there's going to be more blood flowing around in this, so the pressure is going to increase. In the UK in 2016, there were 525,048 deaths. Cancer caused 30.4% of all deaths, and throat cancer caused 5% of the deaths of cancer. So calculate the mean number of people who died from throat cancer per month in 2016. So there's quite a lot to do here. So you've got 525,048. And we want to work out how many people died from cancer as a result of that. So you would times that by 30.4 over 100, and that gives you 159,614.6. All right. Now what you do is you want to find 5% of that for the throat cancers. So for throat cancer, you would do 159,614.6 times 5%, so 5 over 100, which gives you 7980.7. Again, you can't have point something of a person, but I like to just, again, not really round up till the end. Now it says it wants you to work it out per month. So you need to divide this by 12. So 7980.7 divided by 12 gives you 665. Increased methylation of the promoter region of a tumor suppressor gene causes one type of human throat cancer. In this type of throat cancer, cancer cells are able to pass on the increased methylation to daughter cells. The methylation is caused by an enzyme called DNMT. Scientists have found a chemical in green tea called EGCG, a competitive inhibitor of DNMT. EGCG enables daughter cells to produce messenger RNA from the tumor suppressor gene. Suggests how EGCG allows the production of mRNA in daughter cells. As a reminder of how increased methylation works, we have binding of methyl groups to DNA, which makes it less accessible and reduces the activation of RNA polymerase. So that's how DNMT can lead to throat cancer, because it's stopping tumor suppressor genes from being expressed. EGCG inhibits DNMT. So because it's inhibiting DNMT, the tumor suppressor gene can be expressed. You can make RNA, which uses RNA polymerase, and transcriptional factors can actually bind to that region. So you've got to just clarify that. So it says suggest how EGCG prevents this. It's an inhibitor, so it binds to the active site of DNMT. So we're going to say EGCG binds to the active site of DNMT. What does that do? The DNMT can't methylate the tumor suppressor gene region. If it can't do that, that means that RNA polymerase can be activated and the tumor suppressor gene is expressed. With these kinds of questions, it's important to spot the details they give you in the question, especially in this paper, which is synoptic. So yeah, you could have just said like, there's less methylation, so there's an RNA polymerase. It's suggesting how EGCG actually does this, and because it's told you it's an inhibitor, it binds to the active site and so on. The scientists investigated different amounts of EGCG on the growth rate of the throat cancer cells grown in vitro. The results are shown in figure 2, which has been removed due to copyright and restrictions. A reporter who reviewed all of this work concluded that drinking green tea could be a cure for cancer and suggests 3 reasons why this might not be valid. So I'm presuming what they probably had here is the amount of EGCG, like, in green tea and not in green tea and stuff like that. And then they were like, number of cancer cells or whatever in the figure. So you can still tackle this question if you wanted to. This reporter has said that green tea cures all cancers. We've said in the question that EGCG is found in green tea, which may help in throat cancer. So already you can say, well, this has only been tested in throat cancer. It might not work for other types of cancers like lung and prostate, etc. The next thing you could probably say is that, not all cancers are caused by increased methylation as well of a tumor suppressor gene. You can have proto-oncogenes becoming oncogenes, and you can, as a result of mutations. So there's other causes of cancer, not just this. So other causes cancer. Because this journalist is making a claim that drinking green tea could be a cure for all cancers. For those who have cancer not secondary to methylation, that's probably not the case. And as well as that, we do not know how much EGCG is in the green tea, which is also another important thing to talk about. In these sort of questions where it's evaluate or suggest, it's usually a bunch of data from the whole question and somebody making a pretty daft conclusion like this saying, oh yeah, drinking green tea could cure all cancer. In these kinds of questions, you should think about the sample size, the time frame it's been worked in, What's been tested? Was it tested inside a human? Was it tested in cells in vitro? Was it tested in animals? Other things you might want to think about as well. Is it other types of disease or cancer? Or is it just one? Are there any other causes of this disease? So, in this case, methylation isn't the only cause. It could be proto-oncogenes becoming oncogenes and other mutations. Is the amount of medication controllable? Do we know how much is actually in this? Because green tea, you get all kinds of different types. As well as that, does it actually cure it? So in this mark scheme, it says that it only reduces the growth rate. So obviously, I can't tell you, I wouldn't be able to conclude that from the absence of data. But does it actually work? How much does it work? And also, as well as that, have other things been controlled as well? In these kinds of experiments, generally speaking, in exams, if a scientist has done it, then you can assume that proper things have been put in place. I think AQA put guidance out on that. Don't quote me on that. But if a scientist has done it in general, you can assume that the sample size is good, the time frame is good, and the experimental method is good, unless otherwise. Generally speaking, sample sizes, like if you have students doing it, something sample sizes might be a bit whack. But if it's a scientist, the methodology should be assumed to be good, and you need to actually use the data and think about like what's actually going on. Okay, so it says to describe the advantage of the Bohr effect during intense exercise. Whenever you're exercising, cells respire and they produce carbon dioxide. This increases the PCO2 of carbon dioxide in the blood. With hemoglobin, the partial pressure of carbon dioxide can also affect the affinity of hemoglobin for oxygen. If you have a higher PCO2, hemoglobin likes to give up more oxygen more readily, so it has a lower affinity. This is a good way of getting more oxygen to cells that are respiring during intense activity. So that's what this Bohr effect is. So when cells respire, they make more carbon dioxide. This increases PCO2. This then reduces the affinity of hemoglobin, so it increases its unloading and its dissociation. That means in cells with lower oxygen concentrations and higher carbon dioxide concentrations, more oxygen is being released. So that's why it's useful. So the Bohr effect, why is it? It's not telling you to say what the Bohr effect really is. It's just about how useful it is. So what it does is it reduces the affinity. So it reduces. I'm gonna write HB for sure, but you write the whole thing, obviously. Affinity for oxygen. So it dissociates more readily at lower partial pressures of oxygen. But why is this useful? What's the oxygen actually used for? It's for the aerobic respiration of cells. So this is also overlooked as well. Loads of people will talk about how great oxygen is and stuff, but they don't say the obvious. It's saying, why is it good during intense exercise? You need to clarify that it's used in aerobic respiration because you can still release energy through anaerobic respiration, which doesn't need oxygen. So don't forget to include that. Okay, so a cyclist completed a fitness test on an exercise bike. The intensity of the exercise is increased every 10 seconds. The test finished when he was unable to cycle any further. The partial pressure of oxygen, PO2, and of the carbon dioxide, PCO2, in the air breathed out was measured. Figure 3 shows these results. So what have we got going on here? So on this axis, we've got the partial pressure of oxygen, which is a straight line, and you can see it's kind of going up and then down and then back up again. And the amount of CO2 in the air breathed out is kind of just stagnant and it's not really doing very much. Ventilatory threshold, VT, is a measure of the point where anaerobic respiration increases because aerobic respiration can no longer maintain muscle contraction. Okay, so we've got lots and lots of hefty details here. So VT can be identified as the first point when there's an increase in PO2 breathed out without an equivalent increase in PCO2 breathed out. Use figure 3 to determine the time after the exercise started when the cyclist reached VT. And then it says calculate the ratio of PO2 to PCO2 at this time as well. So we're looking for the point where PO2 increases and PCO2 doesn't really increase either. So here it's climbing up and here it's kind of climbing up. Here it's climbing up, it's climbing up. You can see it drops and then it starts to climb back up here again, but there's not really an increase on the outside here. So the time after in the first part is 10 minutes. Now it says to work out the ratio of PO2 to PCO2 at VT. So PO2 to PCO2. So yeah, to work out so each square is each tiny square is 0.4. And one thing to note is that this starts at 8 and this starts at 4 because whoever made this graph wants you to perish in hell, honestly. So we'll start with oxygen. The oxygen's on the left. So that's going to be 10 plus 2 lots of that. So it's going to be 10.8. And then for the PCO2, it's going to be 6. It's going to be 6.4. It's because it's PO2 to PCO2 and it's something to 1. We're going to divide by the 6.4. So divide both of them by 6.4. And that gives you 1.687521. Just make sure to always double check the axes like that because I got caught out on that. An increase in the intensity of exercise produces an increase of the volume of carbon dioxide produced. However, Figure 3 shows that PCO2 in air breathed out did not show a large increase during the exercise. Suggest one physiological change that could cause this result and explain how this physiological change can allow for the removal of the increase in the volume of carbon dioxide produced. So what it's saying is as you exercise more, you're obviously making more CO2, but these results don't show it sort of building up and piling up. Suggest what might be the case. So when you're exercising, you're breathing more, but in each breath, you're removing the similar amount of CO2. But because you're taking more breaths, you're shooting out more of that carbon dioxide. So increased breathing rate, you're breathing fast, so you're taking more breaths in a given time. But there is similar CO2 in each breath. So when muscle fibers have very low concentrations of ATP, they may get ATP in the following ways. So they can use AMPK, an enzyme that oxidizes fatty acids, or they can use phosphocreatine, which donates phosphate to ADP in anaerobic conditions. And here's how they work. So muscle fibers have very low concentrations of ATP. At less than 80% of maximum muscle effort, AMPK is used, but if you have more than 80%, then the phosphocreatine system is used. And AMPK makes hella ATP, and the phosphocreatine system doesn't really make very much. So at more than 80% of maximum muscle effort, ATP can only be made for a limited time. Suggest why. So at more than 80% of maximum muscle effort, we're using phosphocreatine, and that donates phosphate to ADP in muscle fibers. It can only make so much because of all these options, ATP can't move into muscle fibers at a fast enough rate is incorrect because ATP stays in there. Muscle fibers produce too much lactate, that's irrelevant. Muscle fibers quickly run out of ADP, not really, because as you're hydrolyzing ATP, you're making ADP again, so it's gotta be this one. Muscle fibers have a limited amount of phosphocreatine, which is the case. There's only so much you can take, and bodybuilders often take creatine supplements to try and overcome this. Whether they work or not, I'm not too sure. GW1516 is a performance-enhancing drug. It activates AMPK and develops slow muscle fibers at rest. Use Figure 4 to justify why professional athletes are not allowed to take this and do not include details of chemiosmotic theory in your answer. It says here that AMPK is activated and fatty acids are oxidized into acetyl coenzyme A. It says do not talk about chemiosmotic theory. So we're not really supposed to be talking about oxidative phosphorylation here. It also says that you're making more slow muscle fibers as well. Acetyl coenzyme A is what enters the Krebs cycle. So if you've got more acetyl coenzyme A, you've got more Krebs cycle turns, meaning you've got more reduced NAD being made. So we'll say that then. So increased acetyl CoA enters Krebs cycle. That means there's more production of reduced. I'm going to write red for short, but you write the whole thing. Reduced NAD and FAD actually. Therefore, more ATP made. However, that's 3 marks. So we can see that there's something else that needs to be addressed, and obviously slow muscle fibers at rest. Slow muscle fibers are quite good because they can endure more exercise and fatigue less. So increased slow fibers, more endurance. Oh my god, I can never pronounce this word. Erythropoietin. So EPO is a performance-enhancing drug. It can increase the hematocrit, which is the percentage of red blood cells in the blood. A heart attack is caused by a lack of glucose and oxygen being delivered to the heart via the coronary arteries, and the overuse of EPO can increase the risk of a heart attack. Suggest how. So your coronary arteries, they're blood vessels, and you've got like these little red blood cells. And if you've got hella red blood cells, eventually what happens? Blood vessels block. And if the blood vessels block, no supply to the heart attack. So EPO thickens the blood and could block coronary arteries because there's so many of those cells. It can also happen with people who takes testosterone exogenously for bodybuilding purposes. So there's a little thing for your essays. Okay, so the normal hematocrit for human males is 47 plus or minus 5%. For professional male cyclists, the maximum hematocrit is about 50%. The normal hematocrit for human males is 47 plus or minus 5%. Okay, so that can either be 42 or 52. So for professional male cyclists, the maximum allowed is 50%. A student suggests that professional male cyclists should be allowed to use EPO until their hematocrit is 50%. Why is that not valid? Well, some people, if you have a hematocrit, so for example, if you have a hematocrit of 42, you're going to be able to take EPO and get it up to 50. So you've got this hella big advantage. But somebody who's got 50 can't do anything. They can't take anything. So it's just unfair to people who have 50, because it says here it can go as high as 52. So that's just unfair on them. And also, as mentioned above, there's health risks as well. Somebody having a heart attack, probably not a good idea. So some cyclists get a bigger advantage. You get an 8% increase versus like 0. So that's not really fair. And also there's health risks associated with them as well. I'm just gonna write health risks. But yeah, you get the picture. Okay, so now here comes the dreaded essay. Everyone panics on these and is like, "Oh my god, I can't do them." My advice is to start doing essay practice as soon as you can. You don't need to know the entire curriculum to start doing essays. Because if you do them, it helps you reinforce what you know and keep what you know in your memory. So I'd start doing them as early as you possibly can, probably after you finish the first year. So it says the functions of enzymes and their importance in organs. I usually recommend picking about 4 topics, maybe 5. And the way I like to think of it is think about the curriculum from start to finish and what you can talk about. So I like to think of them as you go through the curriculum topics wise. So if we think about things from topic 1, well, you could talk about first of all how proteins how you first talk about how enzymes actually work. So they lower activation energy by providing an alternate reaction route by stressing the bonds, etc. And that's obviously really good because if you're a biological organism, you don't want to absolutely obliterate all your energy trying to break apart some food you ate. That's kind of pointless. So enzymes are quite important there. So you could talk about DNA and its replication. So that's quite important for genetic continuity. So, being able to pass on genes to offspring and continuing a species. ATP. So you can talk about how ATP is hydrolyzed to release energy for movement, for growth and stuff and cellular processes. Without that, you wouldn't be able to do that. And also, if you didn't have ATP synthase, you couldn't regenerate it either. You can talk about the immune system. So topic 2, your immune system. So you can talk about, lysozymes and phagocytes when they engulf and digest pathogens. You can also talk about digestion and absorption itself as well. We need to digest and absorb things to break them down into smaller, absorbable molecules so we can assimilate them and actually make use of them. DNA and protein synthesis. You can talk about protein synthesis and RNA polymerase. You could also talk about um, in genetic diversity and adaptation, you could talk about bacterial antibiotic resistance. So some of them can develop penicillinases, which break down penicillin. Sorry, photosynthesis, come on, that's pretty self explanatory. There's enzymes all the way throughout that and respiration too. Those are obviously massively important because photosynthesis provides food sources for the entire food chain from start to finish. Without photosynthesis on the planet, we wouldn't be able to harness energy from the sun to actually provide food for organisms in trophic levels. And obviously, respiration is massively important for, growth, movement, and all the cellular processes that happen. Synaptic transmission, so you can talk about acetylcholine esterase. If you have too much acetylcholine in your nerve synapses, it will continue to stimulate your nerves and lead to things like muscle fatigue and spasms and stuff. Sorry, muscle contraction as well. You can talk about enzymes in them, controlling blood glucose as well, and even recombinant DNA technology, so DNA ligase, restriction endonucleases. So there's massive amounts of stuff you can talk about. So always just list out topics like that and then pick 4 that you really want to talk about. In all of them, you should talk about how they sort of work vaguely and their importance. So people just list out why, enzymes work, but they never talk about the importance. That's what this essay wants you to talk about. So they want you to say that, for example, all of those things that I just mentioned there. So for example, with ATP, we can synthesize ATP, we can hydrolyze it to release energy for movement and growth. That's very important because it allows animals to escape predators. It allows cellular processes to, be accelerated without having to obliterate all your energy stores trying to break apart maltose, for example. So always talk about the importance as well. In digestion and absorption, that's a pretty good one you could talk about. You want to be able to actually make use of what you've you've eaten, and without enzymes, you'd absorb things too slowly, and you wouldn't be able to develop, grow, and repair tissues and stuff like that. So that's important. So the next one is talking about the causes and importance of variation in diversity in organisms. Now you have to be on God knows what to choose this question over the enzymes one, in my opinion, but you could still do the same principle. So the causes and importance of genetic diversity. So thinking through the topics, in year 1, you can talk about mutations. So mutations can give rise to genetic diversity, and that's good because if you have if you're an agriculture, if you're a farmer and you've got one type of banana that's susceptible to a certain disease, some may have mutations that allow them to survive that disease so they can carry on being produced, or else that farmer's lost all his stock. Meiosis as well. So when organisms are reproducing, similar thing, they all might be susceptible to one disease, but meiosis might allow some organisms to survive. And adaptation as well. So, certain organisms may have better adaptations to survive in a certain environment. So, for example, rabbits with bigger ears have mutations and, genetic changes that allow them to have those bigger ears so they can detect predators earlier and survive and reproduce. You can also talk about biodiversity within a community as well. So food sources, habitats, and niches, why that's important as well. Because if you're a farmer and you've only got one type of crop or one type of, livestock, if one disease could wipe all of them out. What else can you talk about? In muscles, you can talk about slow and fast twitch muscles. They're genetically determined. So that can affect athletes and sort of their abilities to participate in sports and stuff. Inheritance, so there's a lot to do with that. So, for example, inheriting diseases as well. And you can talk about autosomal recessive disorders like cystic fibrosis. You can talk about X-linked stuff, epistacy, lots of stuff there. You can talk about evolution leading to speciation, so allopatric and sympatric speciation, and how that's given rise to all of these different organisms on the planet. You could also talk about things like gene expression in cancer. Obviously, variation can lead to some people being more susceptible to certain types of cancers. So if we're able to identify sort of genetic changes that predispose them, such as BRCA1 in breast cancer, that we can help and identify and screen and treat them earlier. And you can also talk about identifying heritable conditions as well using DNA probes. So there's a lot to talk about. And again, same principle. Again, I would strongly recommend doing essay practice as soon as you finish the first year, really, because at least you can get half of the essay done. I know that's been a very long video, and I know I've gone quite quickly, but thank you very much for watching. Please, like, share, and subscribe, all of that stuff, and share it with your friends.